Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So it's my pleasure to announce Donna Pierre as our colloquium speaker today, and she's an associate professor in the Department of Biological Sciences and Systems Biology at Columbia, although she comes from a similar background to myself, which is machine learning, and has since moved very heavily into making some great advances in understanding biology. And so her lab has been doing a lot of work on interpreting single cell analyses, and she's gonna, I think, speak a lot about that. And this talk is meant, I know there's a lot of people from outside of Microsoft today. The colloquium series is meant to be very accessible to a broad range of people because we have economists and social scientists here. So forgive Donna if she doesn't go into as much detail as you would perhaps like her to, because that's what we've asked her to do.
Yeah, exactly. I don't need this. I think I might put.
Oh yeah, you turn that off.
So thanks a lot. It's a pleasure to be here again. I was told, don't put too much biology, don't put too much computers, just come up and dance. So I'm not a good dancer, so I'll try and nevertheless say something without saying something too heavy. And Jennifer said we're really interested in single cell data and understanding single cells, and we're looking at it from a lot of aspects. And I think one of the most fascinating things that you can ask, and this is something that's fascinated humankind for quite a few hundred years, a picture by Leonardo da Vinci. How do we form from a single cell? As I was talking earlier, and the whole audience heard me, I was talking about my genetic experiment with my husband, and we have two wonderful daughters, and they started from a single cell and developed into this wonderful human being with arms and legs and brain tissue and gut tissue, and it all knows how to create itself off a single cell. And so how does it do that? And generally speaking, our bodies really are composed of a lot of different cell types. And at first, it's very easy. Okay, here's a heart, here's a lung, here's the immune system. But when you drill into each one of these tissues, you see that each of these tissues in their own has multiple cell types, and each of them are very different and creating a very different role. And at this point, we've mapped our genes. We know there's the Human Genome Project. We know every gene in our body. But we're very, very, very far from cataloging every cell type in our body. And some of these cell types do some very important things. And some of these rarer cell types that have been recently discovered are really responsible for a lot of diseases. So it's not enough to know the genes. You actually need to know the cells, where the genes are actually doing their things, where all the action is happening. So even figuring out what type of cells do we have in our body and what are they doing, something that most people would probably think we'd already figured out if you don't know biology. We're far from it. I would say we're 5% into this. It's really pathetic. And so that's, I actually advocate for mapping of the cell types. And one little easy model system to look at is the immune system. And why is the immune system great? It's very accessible. You can hand out your hand and pull out blood without chopping someone up. I mean, if I ask one of you, can you give me a little piece of your kidney or a piece of your heart, you might not really be excited about that idea, but if I ask you for some blood, you might be willing to do so. And blood has a lot of variability, a lot of different cell types that do a lot of important things that keep us healthy. And again, one of the fun things here, there's a lot known, so you're not like going completely blinded into a world where you know nothing about, but there's enough new stuff to learn so that this is a great place to begin trying to look at approaches for understanding cell heterogeneity, for identifying cell types, for looking at how variable they are. And one of the things that you should know, again, I'm supposed to give you enough biological background to understand part of what I'm saying, is so we have these immune cells. They're very different. There's B cells, T cells, monocytes. You might have heard some of these terms. And how do we separate them? They have these markers. The immunologists have figured it out. Each of these cells are coated with special proteins. And most people don't even know what these proteins do, but they know that different protein combinations, like CD whatever, CDX, CD45, CD3, CD19, different combinations of these markers basically are cell identifiers for all we care. They probably have a real biological role, and most of them is unknown, but for our purposes, they're like, the barcode. Okay, so now what we want to do is we want to measure these single cells, measure their protein levels. Here are different single cells. Each little color here is a different protein. For example, these barcode CD surface marker proteins, and we want to measure a lot of single cells and basically treat each single cell as a learning sample and looking at the combination of whatever we measure to sort of study what combinations exist, what are cells doing. If we take single cells and measure each and every one of them, now we have a rich sample. So for this, you really need a technology. And this is what really opened the field up until maybe 5 years ago, particularly, 4 actually. It was really hard to get many dimensions in single cells. So you could get a lot of single cells, but you'd maybe be able to measure 6 dimensions simultaneously in each single cell. And this new technology, which basically changes the way we can label and basically identify these proteins and quantify them, allows us now to get up to 45 dimensions in individual cells. So again, 45 for some people is considered low dimensionality. If you're thinking about the human genome, 20,000 genes, that's nothing. But going from 6 to 45 really does give you a big new world to explore. And again, I want to give you a little bit more background to what people used to do. So you have all these CD markers, and immunologists have figured out cell types. And what they do is they draw gates. They basically draw axes and say, if CD3 is above some threshold, then that's a T cell. If CD19 is below some threshold, it's not. And basically cut out boxes around the data and pull out these cell types through sequential drawing boxes around the data and throwing everything else out. But when you have 45 dimensions measured rather than 6 dimensions measured, this stuff gets really hairy, and this doesn't allow you to discover new stuff. So we want to get an unbiased and complete picture of the entire thing. And to this is a published work by my student or ex-student, Elad, is we're going to go to another field. What we like to do in biology often, I forgot all my machine learning, so yeah.
Sorry, I should have asked this maybe earlier. Is there a ground truth notion of what constitutes a cell type?
So there's a partial ground truth notion. That's the thing. There's a lot of cell types that are known, and there's a little bit fuzziness. This is biology, so it's not math. Yeah. You're not going to get a formal definition. Everything's going to have 20, clauses of exception. But there is some notion. There are some ideas. So that's what I said. The immune system is good. They're not going in blind. There's a fuzzy notion of a large number of cell types, yet a lot of new ones to explore. So it's this nice balance between some stuff known, a lot of stuff to discover. That's the best place to be in if you're sort of venturing to discover a new world.
So
yeah.
Just to take an example to see if I'm understanding the meaning of cell type, if you scraped skin cells off of me and an African-American person and measured pigment levels, I think you would get like very different levels of whatever proteins are involved in pigmentation. So an analysis that plots pigment proteins might say, oh, those are completely different cell types you just analyzed.
Well, people don't use that type of analysis to describe pigment proteins. They might use not the abundance of the pigment, but some other measurements in the cell that define, for instance, the transcription factors that define the cell type. So here, there might be great differences between these immune cells. Take, for example, this is immune cells, so they're probably more similar. They're on the inside. The immune cell of a young person, a T cell, for instance, an effector T cell of a young person and an old person are going to be very different, but they're still going to have that same function, even though there's going to be a lot of differences. Certainly, the T cell between a currently sick person, infected person, and a healthy person are going to be very different, but there's still going to be some shared core functionality between the sick and the healthy one. Okay. So each of these cell types doesn't mean everything is exactly the same. There's a cell type which basically does a same core function, same direction. You hit it with one stimulus and it goes that way, but then there's a huge amount of variability in how it does that. And you want to understand both the cell types and the variability also within each cell type. Those are two sort of interesting questions.
Okay, so the ground truth meaning of cell type has to do with biological function.
Yeah, the idea is you want a cell state that has some type of coherent function, as fuzzy as that's defined. Again, it's not a mathematical definition, but it's a conceptual one. And so we, as linearity is great. It makes things easy, quick, and robust, but no can do. This data is highly nonlinear. It's not Gaussian. All the things we like as mathematicians, out the door in biology. Not if you want to be realistic. And so we sort of hijacked nonlinear dimensionality reduction. I was, introduced as someone who once studied machine learning. I since forgot everything, so I can hijack other people's approaches. In this case, I'm going to hijack Mateen and Hinton's work on TSNE, which is a really great algorithm. And for those of you who don't know what this is, the idea is we're humans. None of us can really see that well in 40 dimensions. Well, maybe some geniuses can. There may be some people like that in the room, but certainly not me. We like 2 dimensions, maybe 3 dimensions. So we want to take this 20, 30, 40 dimensional data and project it down to 2 dimensions so that it preserves as much of the structure and the geometry as possible. So in a little toy example, here we have this curve. And in this projection, we pick up the fact that it's a 1-dimensional curve. We pick up the fact, where the curvature is greater and smaller. All that's preserved in our projection. And even imagine each dot right here is a cell. This is, of course, a toy example. And it's colored by neighborhoods. So cells that were nearby in high dimensions remain nearby in low dimensions. Cells that were far away in high dimension remain far away in low dimension. So basically, the idea is to find such a projection that preserves this as best as possible. And really, we searched for a lot. We tried to develop our own algorithm. And this is when sending your student to NIPS is a great thing. We were stuck on it a while. Elad went. He heard Hinton talk about this TSNE algorithm at a talk at Hinton. He's like, let's try it. And after being stuck for a while, magic. We had to adapt it a bit, particularly it wasn't really well adapted for biological data in terms of normalization. And at the time, it didn't want to run on more than 5,000 cells. So that's a little bit of engineering. But really, the TSNE core optimization function really does magic the biological data. And here's the magic. So what we have here, every little dot here is a different individual cell. And we take its 13-dimensional information and project it down to 2. There's no real meaning to these axes because it's all a pairwise projection. It's meant to preserve some pairwise features between all pairs of cells, or dots for that point. So these axes don't mean much. We could sort of rotate it around, but we basically see the pairwise relations.
Did you say 13 earlier? You said 45.
Oh, there's 45 that can be measured. Here we have 13 measured. What we did in this particular data is we measured 13 surface markers, which are who am I, my barcode. And this was before, this is an old data set, we didn't have 42, we had 31, and we measured 18 internal markers, what am I doing? And so we don't always project on all of them. We typically often do this projection on the who am I surface marker, barcode markers, and that we have 13 measured in this specific case. Now these cells are colored based on expert annotation. So of course the algorithm doesn't know anything about immune cells, doesn't have this classification. This is done independently, and you could see how accurately it really picks up all these big major immune cell types. Every color here is a different cell type. Nearby colors are actually very related cell types. Early B cells, late B cells, it all makes a lot of sense. Yeah?
So how were those 13 markers chosen? Because going to this point, if, for example, one of those markers chosen was, I guess, the skin cell example, like melanin, then the choice of that would influence probably how this projection goes.
So that's where you actually use your biological knowledge. And these were the main markers that help in our, when I showed you earlier those lists where you separate and cut things out based on these threshold tests. So these are the main markers used by immunologists to identify immune cells. And the nice thing is that this algorithm is actually not very sensitive to which ones you choose. So we did a lot of tests where we removed a lot of them and still got most of the structure. You can't, of course, remove all of them. Everything depends on the markers. Obviously, this is where, I don't believe in machine learning can blindly replace a biologist. Some machine learning people would want to do that, but you actually do need a little bit of biological thought going in. Yeah?
Do you consider cell segmentation since you're saying each cell is a sample?
What do you mean? Cell what?
Do you segment the cell? You don't know which area is the cell, right? It can be two cells or, overlapping of cells.
It's there's a lot of efforts to actually make sure that it's a single cell. Of course, there's mistakes, but they're sent one by one through the machine, and we have ways of identifying when two went at the same time using a couple different tests. So of course, there's always mistakes, and real data is dirty, but each of these actually represent an individual cell, and there's a lot of ways to identify when two cells went through the machine at the same time. So each dot here measures a single cell, up to some things we missed. Yeah?
So I guess if we were to include lots of markers that weren't actually useful in distinguishing, how robust would this method be?
Oh, again, it would probably give you something different. It depends on what biology you want to find. So yes, it does depend on what markers you measure. In the melanin example, since melanin is so variable, then melanin might become the strongest signal in your data and might obscure the thing you're more interested in, because you can't preserve everything from 13 and 20 dimension into, and the stronger signals are the ones that reside. And sometimes if you have a very strong signal for something that you're not interested in, it will obscure the thing you are. So again, you have to be smart. This doesn't replace being smart. It's a visual aid. But the nice thing is you look at 2 dimensions and visualize things for many dimensions. And this is what I said earlier. Here, we remove 1 marker and get pretty much the same picture. Here we remove 4 markers. So this is a projection based on only 9 of the markers. And these markers don't say anything to most of you, or probably all of you, but these are the main markers used to separate these cell types. These are the number 1 canonical markers to be able to identify these cell types. And once we removed all 4 critical markers, you see that it's not as clean as here, but even when the 4 most important markers were removed, and there's only 9 markers left, there's still a lot of structure in the data that resembles the full set. So this is actually remarkably robust, or there's a remarkable amount of redundancy in the information coded into these barcode surface markers. Yeah?
So we're looking at 2-dimensional projections of the 13 or whatever.
Yeah.
Is there any reason you can't just do some kind of clustering in the full space? Is that what wasn't working?
That wasn't working for a long while. If I get there at this speed, might not, we'll actually see a clustering. No, we'll definitely get to the clustering. So this is just to show you that it's not linear. This is PCA, and you see that there's some structure. PCA is a powerful thing, but the data is not linear. There's no projection. It sort of pushes and squashes these different cell types together. PCA is certainly not as nice as this. The space is indeed truly nonlinear, and we'll see that in a moment as well. Here's just to show you that some of the other, your favorite nonlinear dimensionality technique, many of them don't work. Many of them are not robust. We tried about 30, and only the t-SNE-based ones work. There's something about t-SNE. Specifically, as I said, this data is nonlinear, and it exists as manifolds in high dimension. And now instead of projecting the stuff onto 2 dimensions, I'm going to project it onto 3 dimensions, just so you can begin to see how nonlinear, non-Gaussian, non-convex these subtypes are. You can see them better separated now in 3 dimensions. They're in the same colors as before, and these are clearly not beautiful, round Gaussian balls as they're often modeled by the community. So the community, 97% of the community likes to model these objects as clean, normal Gaussians. Definitely a realistic assumption here. So what we see is that these immune cell types, yeah?
The CD4 T cells are segmented into 2 very distinct groups. Does that suggest that maybe there are 2 different types of T cells that biologists have not distinguished from each other?
The CD4, is it CD4 and CD8? What is
the pink?
The pink. Yeah, it's probably, I haven't even looked into that, but yes, probably there's two very distinct T cells. I haven't even had, but we're finding more and more structure in the data and surprising things all the time. There's just so much, any one of these things actually to dig into the biological story takes a lot of time. So the reason TSNE was particularly good at working here is when it does look at all pairwise interactions, and that helps a lot in the robustness of it. Doesn't help in the running time. Yeah.
But what does TSNE stand for?
It's t-test, so it's non-normal. So the idea is SNE is stochastic neighbor embedding, and when you do it normally, it projects. You have a crowding effect where you have a lot of things from high dimension, crowd, and low dimension, and then when you try and I didn't go into the full details of this algorithm because that's not. I mean, there's enough papers to read on it, but the idea is, if you want to actually map the regions, you map one in a regular Gaussian distribution and the other in a T distribution with a heavy tail. And this T distribution sort of in the lower dimension pushes things away so they don't clump and crowd together. And so that's slightly changing the distribution to a heavy tail distribution in the lower dimension. And that helps or really allows you to overcome crowding. Otherwise, if you don't use it, everything just like clumps together to the middle. So what we found is that TSNE, and we did a lot of synthetic data, is particularly suited to look for multiple manifolds. So many of these methods are really good when you have only one manifold that they have to map. But if you throw even two manifolds into your data, they get confused. And TSNE, for some magical reason, is able to handle and separate multiple manifolds really well. And what are these manifolds? So what we view it as a biologist is these cells are dictated by a lot of regulatory mechanisms. There's a lot of regulatory circuits inside the cells that are with a lot of feedback and control that are dictating allowed and not allowed states, that are dictating stable states. And this dictates these manifold states that we saw earlier in the earlier slide. And one of the things that we're trying to do now is trying to see the connection between the geometry and the regulation, even though, ask me in a few years, that one is complicated. I just want to give you a few little peeks into what this can do. What we see here is clinical samples. This is actually data generated at a real clinical cancer lab. All these things here, each cell is color-coded by a different healthy person. So there's healthy controls, people that just, as I said, give me please your healthy bone marrow. And you can see that it's well mixed. And there's one patient that has suspect cancer, ALL, leukemia. And that person, their cells are colored in red. And what you see is that there's this distinct region right here that's separated from the rest of the map. And these red cells right here, they're actually the small, tiny fraction of 1 in 10,000 leukemic cells that remained in this patient after chemotherapy treatment. So this is something that takes a very expert oncologist or pathologist that takes a lot of money, and there's a long line for them, a long time to find these cells using conventional methods. And here, if you just map it, because these cells are so different than the normal, they get pushed to a different region, and all of us can see, look, these red cells are mapped aside, and they're not mixed with any of the cells from any of our 10 healthy samples. These are suspect cells. Now we can go and see what are these cells. Are they expressing markers that leukemia expresses? And this is now a method that people have begun to use to actually identify minimal residual disease, which is very important because this way it helps you determine whether a person needs to go to more aggressive therapy or not. Of course, you don't want to make a mistake here. We're using this approach also to identify, this is stem cells in ovarian cancer, this is drug-resistant cells in melanoma. Again, we separate out cells. In these high dimensions, it's sort of hard to see them, but when you map it down, if you have a subset that's really behaving in a distinct way, you suddenly see, oh, these cells are interesting, and then you can see, well, what are these cells? But, back to the clustering questions, I just gave 13 markers, and I just gave these big, coarse separations of subpopulations. If you use more markers and sort of define more subpopulations, then even the TSNE map doesn't really perfectly separate them. This is now a coloring, and I didn't label all the different immune subtypes because these names won't mean anything to you anyway. But as you can see, now that we have more subtypes, these things begin to mix. Every color is a different subtype. And the question is, can we cluster this data? And this is work done by my wonderful graduate student Jacob in collaboration with Erin at the Nolan Lab. Gary Nolan's been my partner in crime in this, and he's been the one to generate this amazing technology that gives us a huge pen of data to play with. And he's the source of candy, or data, as we call it. And so clustering. We tried to cluster. Actually, we spent years figuring out how to cluster this data. And none of the standard approaches work. We don't know how many clusters. They're not of equal size. There can be 3 or 4 orders of magnitude difference in the size of these clusters. They're, of course, I just showed you, they're not Gaussian. They're not convex. They twist and turn. And this sort of cup and ball is very common in the immune system, as we've seen. We've seen this in the data. We needed to really scale to many cells. And we converged on graph-based clustering, sort of social network, community type algorithms, and that suddenly gave us some signal. And so here again, every dot here is a cell, and the idea is we take this high-dimensional geometry that's complicated, and we like mapping it to simple structures. I like graphs, so basically you connect every cell to its k-most nearest cells. This is actually an example of real data. The data is now colored by one of these markers called CD34. It's a early stem cell primitive markers, and you can see these are the stem cells right up here. And you really see a modular structure in these graphs. Of course, this modular structure didn't pop out at us day one. We needed one little trick, one very important trick, and that's the distance metric. Often in a good clustering algorithm, the key is not the algorithm, but the distance metric you use, particularly how we're going to construct this graph. And we spent quite a long time trying to construct this graph. There's a lot of problems, as I said, different orders of magnitudes and size of our subpopulations, different densities in this high-dimensional space. There's spaces that are super dense, super sparse. How do you adjust k? It gave us hell. And the trick that, again, this was Jacob's brilliance. I'm old. I don't have any more good ideas. He thought, well, tell me who your friends are, and I'll tell you who you are. And we basically use the Jacquard method. We build a graph once, and the first time doesn't matter as much once you do the second step. And then you build a graph the second time based on overlapping neighborhoods. So now the edges that you build in the second time are weighed by how many neighbors 2 cells share. And once you do that, it really adjusts for the density. It really makes very what?
So with millions of cells, how do you identify the k nearest neighbors for each cell?
Just with a lot of computational power.
You calculate the million by million pairwise matrix, and then graph operate on that.
Mm-hmm. It's maybe it's not the. I mean, this has not stopped us yet. So, yeah.
So did you start with the Euclidean metric in the 13-dimensional space, or
yeah, we tried a lot of different metrics,
yeah.
and we tried Euclidean, polar, Mahanobus, and all these things. You know, switching around metrics and cleaning outliers and playing with k and adapting k locally. All that improved things a bit for us over like a year and some of work. And then we switched to this Jacquard metric, and then you could do the Euclidean, the polar, the cleaning the outliers, the not cleaning the outliers. Then whatever you do in the first step, as long as it's not crazy, works equivalently well. All the magic is in the second step. So the first step, if you're not doing the second step, can have a huge influence. But once you do that second, tell me who your friends are, I'll tell you who you are, then really you have a lot of leeway. You can do k equals 15, k equals 60. You get the same answer. It's really, that second step is magic, yeah.
So you start with a graph, and then you cluster based on the Jacquard method to tell you who your friends are. So how do you define a friend?
You don't cluster. You rebuild the graph.
You rebuild. So, but how do you define a friend in the first graph?
The first one, as I said, you use Euclid between just the Euclidean metric.
So then you also have the threshold on some. How do you define the initial?
K nearest neighbors.
That's the k. Okay.
That's the k part.
So k is your threshold.
K is a threshold, so it could be very, very. So it's not a threshold on the.
You could also threshold, and then you wouldn't have a K-regular graph. You'd have some kind of...
Yeah, that one actually doesn't work at all.
Okay.
If you want to look for where it fails, that's one of them. You found it. So just to show you the Jacquard metric in action in this toy example motivated by the data, this is what the data often looks like, and there's really no way to disconnect this smaller, rare, but very distinct cell type. Here it constantly connects, and if you sort of change K, change the distance, do whatever thresholding you want, this begins to break up and shatter up, and there's really no way to disconnect it with a sort of nearest neighbor approach that doesn't sort of ask for the Jacquard metric, and the Jacquard metric just splits it up. We just have many, many examples where this just works. You know, sometimes these simple ideas that in retrospect are really dumb and simple, but take a long time to think of, are really worth their magic. And so this is just an animation that my student made of community detection in action. And as I said, we often like to steal things from other people. Take your favorite community detection algorithm. It needs to run on 16 million cells. Take a quick one. Here is every dot here represents an edge. So I have 400 by 400 cells here. There's a dot if there's an edge. Do people see structure here? That's community detection in action. I just really like this animation where this is just reordering the rows and the columns according to the algorithm's output, and voila, you get your modules or your clusters. And how well do we do? So here is our phenograph output. Again, phenograph doesn't know anything except for the data. Here is our annotated output done by an expert immunologist. And not that you have to know what these cell types mean, but as you can see, visually we get it really accurate one after another. This intermediate thing, this black thing here, is very arbitrary. It's a very smooth, gradual, linear line of one marker from here to here, and they just cut it pretty barbarically into 3 rather than into 2. But as you can see, it's quite accurate. Now one can say, okay, this might be an easy problem. So fortunately for us, shortly after we developed this algorithm, or shortly before we developed this algorithm, there was a big competition. A big competition for all methods to identify and label and cluster and classify immune cell types using algorithms. It was run by Nature Biotech, which is the leading computational methods journal. It basically said, calling all people who think your method is good. We didn't have our method ready by then. Put your money where your mouth is. Let's compete and see how well you do. So we have here number 1 and number 2 of this competition that was run late 2013. Compare their performance with phenograph, and if you don't like my perhaps visuals, which you think might hide things, let's use a real quantitative mathematical metric. Here is phenograph switching K from 15 to 60, very, very high F measure, very, very accurate classification. Here is number 1. Here is number 2, and here are some other ones. So as you can see, this really shifts in performance. And remember, I'm now competing against algorithms that were just less than a year ago declared winners in this field. Now, of course, confident on a healthy system really allows you to say, okay, what about cancer? Because in cancer, we have a lot of heterogeneity, and it's both functionally important. There's subpopulations that really help the tumor survive, subpopulations that remain resistant to therapy. And the thing to know about cancer is, and this is something to know a lot about any one of you who gets involved with doing all these big cancer genomic studies, people don't die from, the first therapy not working. Usually, for a large number of cancers, you go to the doctor, you get some chemotherapy, and things look happy for a while. The majority, the bulk of the tumor is eradicated. What really kills you is that small percent, often less than 1%, often less than 0.1% of the tumor, of these really, really nasty cells that can do 2 really nasty things. They can evade therapy, and they can reconstitute the tumor. And it's these cells that are highly uncharacterized in tumors and aren't well known for most tumors, but these are actually the silent killers of cancer. And so taking approaches like these, especially an approach that can really identify rare cell types, we're hoping to be able to identify and classify these rare cell types. And I'm just going to give you a little bit of taste of something we've been doing. We've actually made a lot of progress using this algorithm on really understanding AML, which is another leukemia. We have a big cohort. We use phenograph. And now we use t-SNE to actually visualize each of these clusters. So you see this sort of map of heterogeneity. Here's the healthy cells. And each cluster is colored by the different patient. These numbers are different patients. And as you can see, that even though each patient has its own leukemia, each patient is different, there's a mixture. There's sort of a phenotypic space that AML resides in. There's a lot of heterogeneity. Here's the healthy samples in there. And what you have is actually sort of a blockage in development. So this is early, primitive cells, very, very similar to the stem cell, the cell that regenerates the entire immune system. These are some very late cells right here at the bottom. And here are some intermediate stages. And you see a blockage up here, an expansion. Sort of the development goes wrong. It gets blocked up here. It sort of derails at this area of the map. And we've been studying this quite extensively. Of course, I'm not going to go into all the biology. Just again, to show you an idea of some of the heterogeneity we see now, we have the healthy populations in gray. And now we're putting one patient after another. You see a huge heterogeneity in the maturity of the different subpopulations that reside in their cancer, even though many of them are not quite normal. Here is a cancer which has mostly mature subpopulations, mostly primitive and really derailed subpopulations. You get a lot of variety, but there's also some rules to the game. The way we compare these clusters is, again, we do another second round of phenograph on the cluster. So now we've really identified subtypes, new cell types of AML, which again we've started to understand and characterize. And so now, using this tool, we can begin to dissect the cellular heterogeneity in AML, what cell types exist, what cell types are specific to a specific patient, what cell types are shared, how do they respond to drug, and what are they doing. This is all afforded by such tools, by interrogating this data at single cell level. Yeah?
I think I got a little confused. Are you using phenograph and then from the output of phenograph then running t-SNE on that?
So phenograph, rather than run t-SNE on 16 million cells,
yeah,
I'm using phenograph to identify clear clusters in high dimensional space. I have 600 such clear clusters, about 12 to 28 per cancer. And now I take the cluster medians, and I want to understand the relationship of these clusters both within a cluster, the relations of the heterogeneity within a cancer and between cancers. And that's what this nice map right here is giving me, the heterogeneity within and between clusters, taking 16 million cells, projecting them or mapping them into 600 or so clusters, populations, taking the population means, using t-SNE to sort of map the relations between now these cluster medians. I'm doing another round to try and again, yeah.
And where did this data come from that you're showing us? Is this still CyTOF data?
This is all CyTOF data, yeah.
16 million cells.
16 million cells. So CyTOF runs at 500 cells a second.
Nolan's been busy.
What?
I said Gary's been busy.
Oh, this is a 3-year-old data set and one of the smaller ones. Yeah, no, I think Gary's collected at this point. I don't want to count, but hundreds and hundreds of millions of cells at this point. It's for any of you who deal with single-cell RNA-seq, which I know is bigger at the Broad. The fun thing about CyTOF is, if you're also a machine learning person, n goes up finally. You're not n equals 20, you're n equals 20,000. That's a lot more fun to work with if you want to learn anything. Yeah?
Is it possible to back out which markers contribute to each of the clusters?
Oh yeah, we've done all that. It's
Is it directly from the dimension reduced space, or do you have to go back then and look what are the differentially expressed markers?
Well, looking at the map here, something like this helps you do it, and this map here helps you figure it out from the t-SNE map. But we have methods to do it directly in the high dimensional space, basically to say which are the markers that define this cluster, and that we prefer to do at the cluster high dimensional level. I mean, when we want to do math, we might as well use the full dimensionality. Reason is just for visualization, because our stupid brain is limited to 2 dimensions. But there's no reason not to do the math in the high dimension where you have all the information and you lose none of it.
If you switch the dimensionality and you have way fewer cells or way fewer samples, but way more features. So if you have thousands of features, but 40 or 1,000 samples, would this method still be able to capture those?
40 samples is on the low side, but we've used this on single-cell RNA-seq data where you had a couple RNA-seq and 500 cells, and we've actually found some very distinct structures with this. So I don't know if we'd find much structure with 40. That end is a little bit too low, but a couple hundred definitely did that, and it succeeded. So now I want to do something completely different. I want to get some of it still done today, and that's, so up to now we've been really harshly separating them and calling these separated sets. But let's take these B cells that go from the stem cell to the mature cell and see, is it really, should we really separate it into these, what did I just do? Build my thing. These distinct sets. So we want to trace the B cells. These are, again, I told you immunology is studied, so these are the steps they go. B cells are really, really important. They help you identify nasty stuff that's not self and that the immune system should mount against. And it goes through these steps. Throughout these steps, some cool stuff happens inside B cells, such as the DNA gets shuffled, so it will be able to identify particular antigens from particular intruders into the body. And we want to try and study this process and understand what happens during this process. And one of the fun things about being able to do this is something like the immune system is constantly regenerating in our body. So even though development and the process of going from a stem cell to a mature B cell is a dynamic process, if we take one single sample from our bone marrow, in that same single sample, because it's an asynchronous process, we will have every one of these stages represented. So we could pick up the entire dynamic and the entire all developmental stages and intermediaries just from a single sample. So this is a lot of fun. Only have to, bone marrow samples are painful, I've been told. I haven't tried, so poking them once is a good example. So the way we've been doing things now, the way immunologists have been doing things now, is let's categorize, let's cut out these things, and let's identify cell types. In the B cell case, they're called pre-B cells, pro-B cells. The names don't matter. It matters that we gate them out, we cut them out, we've identified them. And we opened the immunology textbooks. We looked at our high-dimensional data. This is a bone marrow with a lot of B cell markers. It's a slightly different data set than what we were looking at before. And when we opened the textbook and did what the textbook told us to do, and said, okay, how many cells are left over? How many cells weren't put into any bin? 60% of the cells were thrown into the trash. So what we know how to put into a bin is 40% of the cells, and 60% of the cells are unknown. And our theory was, well, this is actually a continuous process, and there's a lot going on in these saddle points, and a cell doesn't turn into a pre-B cell from a pro-B cell at the snap of the finger. It's a process. There's a lot of transitional populations, a lot of things to be learned, and when you just cut out these very harsh gates, you chuck out all these transitional things. So we thought, okay, let's look at the development as this continuous process. And so our new computational goal, and this is something that we can find very easily, we have lots of single cells, we have markers measured for these cells. Can we organize them in a temporal order, basically index sort the cells so that we put them in their maturity or in their developmental continuum, and we'll make the basic assumption that we sort of know where the starting point is, what a stem cell looks like, and markers, these surface markers that we're using to identify these cells, they're supposed to rise and fall in a gradual, continuous manner, or, and they don't just change like that. And once we have this ability to sort the cells, now we can pinpoint all sorts of important events, such as when they burst and proliferate and make more of them, and when that recombination that I told you about, where the DNA shuffles and changes its identity, when all these good things happen, which, by the way, up until recently, many of these events were not pinpointed. The people could not find where this important event of reshuffling of the DNA occurred during the process of development. So again, I just want to show you again this thing which Jennifer said causes her motion sickness, just because I want her to get motion sickness. Focus on the green cells, and you can see how this thing twists and turns. I want you to particularly focus on this thing right here. It goes in, out. This thing sort of really twists and turns. This thing, development goes from here like that. It's, again, the point is you have high degree of nonlinearity. And this nonlinearity is what makes it hard. So this twisting and turning, which I showed you earlier, this is developmental progression just in 2 markers. It goes out here and then snaps back and goes up. So these 2 cells can appear pretty close to each other, even though they're far in their temporal time. Now add on to it the noise in this technology. One thing that I sort of haven't been throwing out very much is this technology is noisy, as most biological data is. It's not pretty like this. It looks more like that. And when it looks more like that, it's a lot harder to separate and say, well, this cell that belongs to this part right here could be really close to here. You get all these shortcuts. It becomes very, very hard to trace this nice red line when your data looks like that. So again, we really found that this neighbor graph is taking this complicated, noisy, high-dimensional data and mapping it to this nice nearest neighbor graph structure really helps find structure, really helps clean up the noise, really helps us go around the noise. And now, because we trust nearby distances, we trust that these 2 very, very, very similar cells are probably at a similar maturity, we can try and get to a farther cell using a walk in the graph. So we trust these neighbors, and if we want to go farther, we walk. But these shortcuts are our bane, and so therefore we developed an algorithm. Again, this was developed by Elad, and this was published in Cell. And we have the Cell paper is really biological, but then we have a 30-page supplement, which is written to a computational audience with lots of nitty-gritty. And basically, we use a favorite trick, something I learned in my master's. One way to fight noise is feed it its own thing, randomness. And rather than taking a harsh KNN graph, we build a KLNN graph. We take K nearest neighbors and uniformly sample from it lots of different graphs where we only subsample L of these K neighbors. So we would hope that if there is a shortcut, we'd be able to, it won't be in most graphs because we're taking, it's very hard an adversary can always stick a shortcut, but if we're taking like K equals 30 and sampling only 5, and the shortcuts are a few, we could hope to avoid it. The other thing we do, which really helps, is again, each of these errors accumulate. Each time we take another step and a misstep, then these missteps are additive. So the farther away you go, the less accurate you are. And therefore, we add random waypoints. These waypoints help us reorient ourselves. So we could calculate the distance not only from the starting point, but from all these waypoints, and having these nearby waypoints to sort of reorient ourselves, to look at distances over shorter distances where they're more trustworthy. Why am I doing this all the time? This really helps us. And with these 2 tricks, the waypoints and the random graphs, we can really, now these waypoints, again, we now do a weighted average over all these waypoints where nearer waypoints, because they're more reliable, way more. And we could take each point and use these waypoints to sort of place it where it belongs based on how the starting point in each waypoint sees it in terms of distance. And you continue to iterate through that and then average over the graphs, and that gives you your final trajectory. So these 2 randomization steps, random waypoints, random subsampled graphs, really give you a very robust trajectory. And then if you have a trajectory which is time, you can average in sliding windows over the cells and look at how some of the different markers, both the ones you built based on and some other ones that you measured for function and activity, change during development. Now this thing is incredibly robust. Here is synthetic data, and we're adding noise. This noise is about 50% of the magnitude of the data itself. And it still picks up some pretty good data. This is like the degree of noise right here. Here we're forcefully adding short circuits. Here we're adding as many as 1,000 short circuits. Here we're adding short circuits that are quite long and far-reaching. And you really need a very large number of very far-reaching short circuits to mess it up. This thing is very robust. Here's the biological answer. And this is the biological answer, which for anyone who's an immunologist could be very, very happy about it. It really matches a lot of known biology. It reproduces across 7 individuals. So these 7 individuals had different breakfasts and saw different pathogens, are very different, and yet we get the same canonical progression. The only thing that really confused people is some of these markers that are really important for B cells, CD10 and CD19, went up later than the biologists thought. But other than that, the biologists were really happy with this. Here's our the way that we're reproducing over 4 different marrows. Each color here is a completely different individual collected on a completely different day. And you can see that the same patterns, the same behavior of these markers are picked up. Here is, we don't really need to start in the correct point. Here we restart from 1/3 the way in, and it's able to correct itself and figure out that we didn't quite start at the beginning. And if we start at the end, it actually just gets the reverse. So we can start at the end and go back to the beginning. We can remove markers, almost any single markers, and get 98% or typically 99% match. The worst one was 92. Oh, if you remove 6, you get 92% correlation. So again, this thing is really robust. And as I told you, this recombination is something that was never pinpointed. So we hoped, again, it's really fun to not just do these algorithms and show how robust it is, but to show at the end of the day, for all your work, you can learn something new. And our goal was to figure out where this recombination happens. It hasn't been found till we could find it. And our algorithm said, well, we could pinpoint it. We measured the enzymes that do the recombination, and we could map the enzymes onto our surface markers and onto our trajectory. And what our trajectory said is this, remember I said the CD10 and CD19 went up late? We said no, it happens before that. And so we could actually use our trajectory to pinpoint exactly when this happens, tell the immunologist where they need to sort and separate their cells to find this recombination. And our prediction was validated. And the reason we succeeded and everyone else failed is the dogma in the field was this happens after CD10 and CD19 goes up. And remember, those markers don't mean anything except for the fact that the immunologist never bothered to look before those 2 markers because obviously it happens after those markers. But when you tell a computer who doesn't know anything about the immune system, give me the unbiased, data-driven answer, the computer says, guys, you should be looking before CD10, CD19. You'll find it now. And we managed to find it. So now this has been very, very, very accurately pinpointed in 3 different new subpopulations that were previously not known. Now, I wanted to talk to you. So our 3 populations are called POP 3, 2, 3, and 4. And I wanted to show you just how rare they were. So this is the sort of pie chart of the entire immune system. And these are primitive cells. These are early cells. They're a tiny slice of the pie, this red slice right here. We're going to expand it to these red primitive cells. Our population 3, where this recombination happens, is 7 in 10,000 cells. And our algorithm was so accurate that reproducibly across 7 marrows, we managed to place these 7 in 10,000 cells next to each other accurately in a single bin so that we could later recognize them as a place where interesting, cool stuff happens, both this recombination I told you about as well as some really interesting regulatory checkpoint behavior that makes sure that these B cells are recombining properly. This population 3, the 7 in 10,000 cells, has regulatory networks that weren't discovered before that are in charge of telling the B cell this recombination happens successfully, go forward, or die. And something like shuffling your DNA, you really want to make sure you did that successfully, because when you don't do that successfully, you get ALL. And we later have some very strong evidence, this isn't proof yet, that a lot of leukemia, B cell ALL leukemias are exactly expansions from this population 3 that that this checkpoint of the recombination happening failed. Yes?
Yeah, so in that sense, the trajectories that you identified, did they have to be like non-branching?
So this algorithm assumes non-branching. We're working and almost completed an algorithm that can branch. So just to get this non-linearity working very well, this robustly, was quite a feat. And this was and one of the reasons we did B cells is because adding a branch onto it is all the more complicated. We now have an algorithm that finds branching. It's not easy. You lose a little bit in the accuracy, so our branching-based algorithm cannot get that same pristine 7 in 10,000 resolution that we can get in a non-branching, but we can certainly now have an algorithm that identifies branching. This is sort of brand new, something that finally is working 2 weeks ago in the lab. So the next thing we want to do is really map the entire body, just like the worm. Like we have this little tiny worm is fully mapped. We want to be able to map all our cell types and do these branching trajectories and be able to do them even more accurately than we can do them as of 2 weeks ago, and really understand how healthy development happens, what goes wrong in disease, how we could use this understanding of development to do regeneration, and all these other fun things. So I hope I convinced you that single cell data is really cool and fun and really gives you a great understanding of cell systems, and taking a geometric viewpoint, both trying to look at this high dimensional space, mapping it down with tools like VSNAN or TSNAN, nonlinear dimensionality reduction, but even more so, because this data is so noisy and complex yet modular and with structure, this graph-based approach of taking the geometry and mapping it to a more robust, more manipulable nearest neighbor graph is a really great way to play with this data. And so finally, these are the two guys that did most of what I told you about, Elad and Jacob. A couple other people in the lab helped. Close collaboration with Gary's lab and the three musketeers there, Sean, Aaron, and Kara, and thank you for your attention.
So if you wanted to do sort of the second half of your talk, but on maybe a cancer cell, so if you wanted to sort of find out the chronology of a cancer cell and its origin, could you use that could you make one of those sort of trajectories?
So for that particular question, we had that idea, and the answer is no. And the answer is not because of any deficiency in the method, but deficiency in the data. What happens is because the cancer is an evolutionary process and survival of the fittest, so let's say you started a healthy population and sort of diverged from that, you're sampling the cancer at some later point, whenever it's diagnosed. Many of the early populations of the early progression of the cancer as it began barren are long gone or long unfit. So what you have is a population and then something at a much later stage in evolution, cutting out the entire in the middle. And what we seem to see, and we've actually looked at some, you could take a model where you actually, in mice, not in humans, trace it a little bit more sequentially, and you can see things. And because of these nonlinearities, let's say you have a point, the closest point to the healthy system when you don't have all these intermediaries to connect and you have this nonlinear curvy thing, really you can't look at closest distance. What you can do is you can, for instance, I showed you that the AML is development gone wrong. So within the cells that you have in the cancer samples, to see the developmental trajectories that still exist in that sample, both as far as where is the cancer developing in terms of evolution of new clones, and in terms of how cancer is a development gone awry. Many cancers are basically the real developmental disease. Those things you can do, particularly once we get the branching working even better. But connecting back to the early healthy cells when that entire chunk is chucked out and not there anymore is not with all the twisting and turning that the cancer does to this space.
So in order for CyTOF and Bisney analysis to kind of transition into the clinical setting, it has to be able to deal with batch effects between different runs of the gene and different sensitivities and different availabilities of antibody binding and such. Is that an issue with Bisney, or if you put in samples from 10 different patients taken on 10 different days, are you going to segregate out the 10 patients instead of the 10 cell types that you actually care about?
So a few things regarding that. Number one, the clinical example I showed was actually on normal flow cytometry data, not mass cytometry data. Two, CyTOF does have quite a few tools, such as bead normalization, which help a lot in the normalization. they're not perfect, I agree with you, but it helps a lot. Specifically regarding your question, what we see in healthy populations and also in cancer, you see the shift in the batch effect. So the batch effect would be, if this is a cell population of a particular type, and here's the same cell population in this batch affected second sample, you'd see like a shift like this. So they wouldn't be like this, they'd be like this. On the other hand, you wouldn't have, batch 1, batch 2, batch 3, batch 4. You would see this sort of overlap of the same cell type hitting the same region with minor shifts rather than, losing the structure entirely is where things stand. Now, of course, it all depends on how big your batch effect is, but in normal batch effects, that's what we see, these minor shifts, but basically the structure remains the same.